Oh, okay. There's just 3 panelists, no logo. Good to know. Thank you. All right. It's 5 after the hour. I imagine people will continue to join us, but let's get going. Hi, everybody. I'm Erin McNabb, Associate Director of Alumni and Parent Relations at Harvey Mudd College, and a proud alumna of the college, class of 2006. Firstly, we hope that you and your families are all doing well and staying safe. It's made me so happy to see all of, as you've registered, all the faculty and staff from campus, our alumni and parents from around the world, and our admitted students and families who are just joining our community. I'm glad we can be here together, even if it is just virtually. This is actually our first Mudd Talks event in an ever-growing series of webinars. So fingers crossed everything goes okay. I may freeze on occasion. This event is being recorded and will be shared later. We will also try to compile articles that we mention or reading that is recommended that we'll send to registrants after the event. The topic of COVID-19 is so wide and varied with new information out every day. So we'll be doing our best to cover as many topics of interest of the audience while also giving some general overview and highlighting our experts' fields of study. Please use the Q&A feature at the bottom of the screen to submit questions during the event, or the chat. A few of you have figured out how to chat our panelists with questions as well. So that is it for my general information. I'm going to hand this over to our 2 presenters today, Karl Haushalter and Nadia Abuelezam.
Great. Well, thank you, Erin. Thank you so much for organizing this, and thank you to everyone for showing up. It's so wonderful to have the Harvey Mudd community here together. Like now more than ever, I appreciate the degree to which Harvey Mudd is more than just a bunch of buildings. It really is this people and this incredible network of people and the relationships that we've formed. Which is why I'm so incredibly excited to have with us tonight Nadia. And so Nadia, it's so great to see you. You took my class on HIV and AIDS in the spring of 2008. It was the very first offering of the class. You did pretty well, as I recall. And I wonder if you could just tell the folks on the call like what you've done since then.
Sure. So I'm so excited to be here. It's really wonderful to be able to do this with Harvey Mudd. Harvey Mudd holds such a fond place in my heart. And so I'm just so thankful to be here. And I'm really grateful to be here with, I'm going to keep calling him Professor Haushalter because I cannot call him Karl for the life of me. So I'm really happy to be here with Professor Haushalter. I graduated in 2009 with a degree in mathematical biology from Harvey Mudd. And I went off to Harvard University, where I was actually able to complete my doctorate in epidemiology at the School of Public Health there. And I wrote a doctoral dissertation on HIV and AIDS, very much inspired by the course I took with Professor Haushalter.
Sounds familiar, yes.
And I am now, after doing a postdoc at Harvard, I'm actually now an assistant professor at Boston College. I'm in the School of Nursing, and I'm part of the global public health program that's growing at now. So it's been really wonderful to be able to go back to education, and I'm also teaching at a college now, which is a huge joy in my life. And I'm just very grateful to be here. How have things been over at Harvey Mudd, Professor Haushalter? What have I been missing out on?
Well, let's see. Well, first of all, if you're going to keep on calling me Professor Haushalter, I'm going to call you Professor Abuelezam. So
no.
So fair is fair. Since you've left the college, I've gotten to offer the AIDS course 10 more times, and so a few hundred students have taken it, including the last offering. We actually did an offering of the course inside of a men's level 2 security prison out of Norco, California, as part of the Inside Out project. And so I took a group of students from the Claremont colleges, including some Harvey Mudd students, and did the class with a mixed group with also a bunch of incarcerated college students as well. So the class that you started with just 6 people, it's kind of like blown up into this whole thing. So I'm grateful for your help with that. Recently, along with all of my colleagues at Harvey Mudd, instruction has moved online, and so that, which comes with all sorts of challenges, as I'm sure you're experiencing in your role too. I teach biochemistry in the spring, so I've taken this opportunity to refocus the last third of the class on the novel coronavirus and trying to understand some of its molecular details, just to give the students a sense of what is this thing that's putting us through such a difficult time.
Yeah, I hear you on the disruption in the classroom in terms of trying to go online, but it's also been really neat to see how the coronavirus pandemic really intersects with so many different topics that we interact with in the classroom. I'm currently teaching a graduate level epidemiology class, which has essentially turned into a graduate level class on coronavirus. And I also teach an undergraduate global public health class. And it's been really, I think, really interesting to see how what's been going on in the world intersects with what we're talking about in class. So it's been a challenge to make the transition, but I think it's been really rewarding as well.
Yeah, and indeed, and really the remarkable thing is, although I happen to have the privilege of being here, like a whole number of my colleagues from essentially every department could talk about the way that their discipline can shine light on this. I was just curious, like, so in your classes, so as an epidemiologist, like what are you kind of most intrigued by, most motivated to kind of learn more about? Like what really is the part of this current pandemic that has caught your eye the most?
Yeah, I mean, I think one thing that I've been thinking about a lot with regards to the coronavirus is, unfortunately, a lot of epidemiologists and a lot of folks in public health sort of knew that something like this was coming. They sort of had anticipated that there was going to be a virus that was going to cause a pandemic of this size. And one thing that I think has been really striking is our ability to respond quickly and swiftly to this, right? So thinking about the reasons why our response may not have been so quick, but then also thinking about the ways in which science has really stepped up to be creative and to be responsive despite perhaps limited resources or despite a non-national response in some ways to the epidemic. So I think there are so many different interesting aspects, but one thing that's really struck me is we've sort of known this has been coming for a while. And so seeing the scientific community step up in many ways has been really exciting to see the responses, the creativity that's arisen in not only the epidemiological community, but also in the scientific community as well. And maybe in similar ways, and I'm sure we'll have a chance to chat, to what happened in the early days of the HIV epidemic as well, with regards to the scientific response. And I know, there's so many different questions that have come up in the past few weeks with regards to coronavirus. And I know many people had submitted sort of a bunch of questions before we started this. And one thing I wanted to ask you, Professor Haushalter, and your expertise and your experience was sort of with regards to immunity. What is our understanding of immunity of this particular virus? What might we be able to expect as we're moving forward in this pandemic?
Yeah, no, that's great, and it's an excellent issue to raise. Sort of following the questions as they were coming in this week, like that was definitely one of the ones that has come up a lot, because as we think about, what does the world look like as we ease up on some of these social distancing, this is really the key to understand what could happen and how fast and how. And I wish I had more concrete and solid answers. And there's a lot of question marks, and there's some educated guesses. When we talk about immunity, this refers to the fact that for many viruses, when you're exposed to it a first time, you develop an immune response to that. If you're exposed to it again, you have a stronger, more immediate immune response, which in some cases can prevent that illness from returning completely. And it's not really a binary thing. And so it's not that you either have immunity or you don't. You can have it in degrees. And so sometimes someone might be able to completely block an infection. For measles appears to work this way. Other illnesses, even though you still get sick, your symptoms might be less, the duration might be less. And so it's not as severe of an illness because of pre-existing immunity. So there's a question of like, okay, for coronavirus, and what does it mean? So if you've been exposed to it, if you've had coronavirus, how protected are you? And then also, for how long are you protected? Is this going to be a durable response? Is it something that's going to fade over weeks, over months, years, decades? So really, we won't know until enough people who've recovered have been re-exposed, and then we can do some, modeling and statistics, and then figure that extent out. And so that's when we actually will get to know. The early clues we can look at, though, are markers of immunity, markers for things like antibodies that are present in individuals who have been exposed to the virus. And antibodies are these proteins that are secreted by B cells. They are part of the adaptive immune response to viruses. And the presence of the antibodies is perhaps suggestive of the ability to fight off that infection later on. Although we don't know at this moment to what extent those antibodies are going to be effective and be neutralizing antibodies that will actually block the virus. There are other markers we can use, like T cell responses and so on, for people who've been exposed. And so looking at people like now with this current epidemic who've been exposed and watching, just doing blood work, we can get some hints at how we might predict they might be immune. We can also look at other viruses. And when we were preparing for this talk, you sent me a really nice article written by one of your mentors at Harvard, sort of looking at other coronaviruses of SARS and MERS. And it seems promising that there will be at least some immunity, and for some amount of time, a year, 2 years, they can find antibodies in people who've recovered from those illnesses some number of years later. But it's right now it is still a huge question mark.
I think one of the reasons I'm so interested in immunity is because I'm a mathematical modeler, so I use a lot of the numbers that are coming out from the science to inform models. And one of the big parameters that we have very little information about right now is how long does immunity last? And that's really going to help us better predict what will happen in the future, which I know is sort of the question of the hour. Is what is this going to look like in a few months? And for any model, you always want the best and most accurate information to populate that model with in order to get really good information out on the other end of the model. So I know that that's one of the parameters that we've been really curious about, and I know we have limited information about, but that's true for many of the parameters that we have right now.
Yeah, and that's a nice segue into something that Erin raised when we were talking with her earlier this week. Is how does someone who's not a mathematical modeler assess how good of a model it is? Because we almost every day it seems we read about a different model, and so in our conversation with Erin earlier this week, we talked some about, well, how can you tell what is a good model, a reliable model, a model that they can hang your hat on versus one that perhaps is less valuable?
Yeah, it's a really important question. I think it's a really tough question to answer. But I think one thing to remember, and I just want to say this up front, is we really actually didn't need a model to tell us that something bad was going to happen in the United States, right? I mean, we could look at the experience in China, we could look at the experience in Italy and know that something was going to happen in the United States. So we didn't really need a model to tell us that. But I do think that there is sort of some need for modeling to help us with the long-term consequences and the long-term impacts, and also then to do some perhaps experiments with different interventions we could put into place to help us contain the spread of the coronavirus. So I think when I'm reading a paper, the first thing that I look at is at the assumptions that the model is posing. So every mathematical model has assumptions. The reason we have assumptions is because a model is really trying to be a simplified version of reality. And so you need to have simplifications in order to represent something with mathematics or with simulation. And so those assumptions are really critical to our understanding of how valid the model results are. Another thing I look at is the data that goes into a model. So there is a phrase that says garbage in, garbage out. So the better the data is that goes into the model, that motivates the model, the parameters of the model, the better the predictions are likely to be on the other end. And then I will say that I never trust a model that doesn't give you some sort of uncertainty bounds in the estimates that it produces. I'm wary of any modeler that thinks they can tell me the exact number of cases of coronavirus at a particular time. So I think being able to estimate and quantify the uncertainty in estimates is really quite important. So I'm always looking at those bounds on the uncertainty when I'm thinking about modeling results. I have much more to say on this. I'm actually trying to get a paper published on this soon. So I think there's more to say, but those are the big three things that I would say I would look at if I'm reading a mathematical modeling paper in the literature.
Awesome.
I think kind of.
I guess, I mean, oh, go
sorry.
ahead, Erin.
We got a question about, are there some parameters like in a model that there's very little information and therefore a lot of assumptions made, specifically about coronavirus maybe, or ones that you know are quite accurate, parameters specifically in models that you know are more accurate?
Sure. So I think right now it's safe to say that at the beginning of the epidemic, I would say the parameters that were hardest to estimate were probably behavioral. So knowing how many contacts people had, how risky those contacts were, that's always really difficult to assess at the beginning. Now, if we assume that people are actually socially distancing and actually self-isolating, you could actually make some pretty legitimate assumptions about people's behavior and the number of interactions that they're having on a daily basis. I think now the uncertainty lies really in the initialization of a lot of these models. And that has to do with the fact that we really don't know how many people really have or have had coronavirus in the United States over the past 2 months. And so we're initializing these models with quite a bit of uncertainty because the data we have is delayed. It's testing data, it's incomplete testing data, and it's delayed. So it's really hard to initialize these models with a lot of certainty. And then the other big parameter that we already mentioned is the immunity, the duration of immunity on the back end. I think a lot of people are assuming that the virus gives you some form of immunity. I think the estimates I've seen are on the order of at least 6 months, but that is a huge assumption. We don't actually have scientific evidence for that quite yet. And so that's another big parameter that I'm a little bit uncertain about, but that really drives our predictions, especially when we think about the late summer, early fall predictions that are really important right now.
I mean, as I think about scientists communicating with the public about our work, it feels like uncertainty is like one of the hardest concepts. Like as a scientist, I think I've discovered that I'm very comfortable with a certain amount of those bounds that you talked about, and that ends up being a hard concept to communicate to the public, who I think often assumes that as a scientist, when we put on our scientist hat, we speak in certitudes, which is not at all, as you know, how we work.
Yeah, it's true. And that's actually something that I've been struggling with over the past few weeks is how do you communicate that uncertainty to the media? How do you communicate that uncertainty to the public and still, maintain the public's trust in science? And I think that that's a big struggle right now is we want the public to maintain their trust in the science, but it's hard to do that when we keep saying, I don't know. I don't know the answer to that question. But I think that that's also a sign of good science is admitting what you do and do not know. And I think that that's actually a strength that we have, being able to decipher what it is we know and not know.
Yes.
I guess another thing that I'm really curious about, and I know you've been working on this for another virus, but another thing that people have been talking quite a bit about is a potential vaccine for coronavirus. And I know you've done some work on some elements of vaccine development for HIV, and I was hoping you might be able to talk about some of the challenges to vaccine development, whether that's specific to coronavirus or in general. I think that might be helpful.
Right. Yeah. Thank you. And vaccine development relies on our ability to determine what makes a good immune response. And so what does a protective immune response look like? And then how do we trigger that in an individual by exposing them to something? And in the case of coronavirus, it's hard to keep track of how many different things are being tried that are different aspects of the virus that might trigger an immune response in an individual whom you give this to, and then figuring out if that's going to be protective. And so I think one of the questions that go into this I think, and I know people are really impatient for the vaccine, and I think what a lot of the slow step for this is going to end up being testing, because in order to know whether a vaccine works, you have to give it to individuals, find out if it's safe, first of all, and that's done with a small number of individuals. And then beyond that, like, is it actually efficacious? And that requires people to have been exposed to the virus, and then for us to see a difference between a placebo and the vaccine that we get, so we know whether it's efficacious. And it takes time to accumulate that exposure. And so as you know, even though we're going at like record-setting speed, I think the previous record for a vaccine was a 4-year development timeline, and we're now talking like 12 to 18 months. The reason it's still that long, and again, that may turn out to be an optimistic projection, is this time to test. There's some ethically challenging and things that I have a problem with about thinking about, could we do an actual human challenge test and sort of to speed this up? And some people have suggested, personally, I'm not in favor of us doing something like that, which means it's going to take time. The good news is, with respect to coronavirus compared to HIV, which is the other virus that I spent a lot of time studying, it doesn't appear to mutate at nearly the same rate. Like, one of the reasons why we don't yet have an efficacious HIV vaccine is that HIV mutates. And so you're not trying to stop one virus, you're trying to stop a whole quasi-species of millions of different viruses that are each subtly different enough that anything that you might develop wouldn't end up being effective. And so, the good news about coronavirus is it does not have that high mutation rate. Another trick that HIV uses is it is literally a wolf in sheep's clothing, where it takes carbohydrates, these sugars, and makes a glycan shield that covers it, which then blocks the action of our antibodies to be able to bind to the protein part of the virus. And the coronavirus does have a number of glycans, but there are gaps, there are holes. You can look at the structure and see where it's likely an antibody may be effective. And so I'm hopeful. It seems like a much easier task than HIV, but no matter how easy it is, it's still going to take time.
Absolutely. And I think, that also goes back to making sure that the public and everyone still maintains their trust in science, despite the length of time that it takes to produce a vaccine. Because right now it does seem
Did Nadia freeze?
It appears that Nadia is frozen.
From an outsider's view that the vaccine is sort of the. I've probably come back now.
Yes.
Okay.
Yeah, you're right.
Okay. I think people are really interested in a vaccine because I think it feels like that is really the only way out of this, which I'm not sure I agree with that statement. I think it will certainly help. But I think that's one reason that the public is so interested in vaccine development.
Can I jump in a little here? People keep talking about herd immunity, and I know it's been thrown around on the news a lot. So can you talk about vaccines and herd immunity to COVID-19?
Sure. So herd immunity is actually a term that we, as epidemiologists, use exclusively with regards to vaccination. So you want to vaccinate a population to a particular rate such that the people who aren't vaccinated are still protected by that vaccine. And so why might not everyone get the vaccine? Well, there are a lot of immunocompromised people who can't actually receive vaccines. And so we want to get that vaccination rate up high so that we build that herd immunity. What's being discussed now is what I would argue a slightly different type of herd immunity, which is this idea of let's get enough people infected such that we don't see as much transmission. And I just want to emphasize that those are two very different things. One is about vaccination and one is about infection. So I don't know very many epidemiologists who would advocate for the herd immunity with infection. We always try to prevent infection. We try to prevent death. There has been a lot of discussion that that might be the way out of this, is to get to a point where we have reduced the basic reproductive number, the potential for transmission under 1, and that might be through herd immunity. And so, a vaccine will help us get there much faster and much safer than getting everyone else infected. But that certainly is one way to reduce transmissions and to ease some of the restrictions. I would not necessarily advocate for that, but that is, epidemiologically, that's probably a valid statement.
Yeah, it was interesting to read today's New York Times and the reports on the antibody test showing that perhaps something like 20% of New York City has had coronavirus at some point. And I wonder, what do you make of those reports on the antibody tests?
Yeah, so there's been a number of tests that have come out in the past few days. New York City, there were 2 in California, one that was leaked from Los Angeles County, and then another one that was a preprint from Santa Clara County. And the Santa Clara County sero survey resulted in about a 2% prevalence of the antibody in Northern California. And as you've mentioned, the New York City sero survey resulted in some areas well over 20% prevalence. And so I just want to say a few things about those surveys, the sero surveys, but also the antibody test. So as we know, the antibody test relies on an antibody response, right? And so the level of response and the level of detectable antibody can actually differ between individuals and differ within an individual over time since infection. And so our ability to check that antibody is going to actually vary between and within individuals. And so the sensitivity and the specificity of these antibody tests is still in question. As many of you may have heard, the FDA accelerated the production of these antibody tests. And so they haven't been validated in the same ways that prior tests have been validated. So we actually don't know how reliable these tests are in reality. So I guess that's a long-winded answer to say that I am not going to dispute sort of the exact numbers that are coming out, but I will say that the way in which these sero surveys were done, there are some issues. So thinking about trying to assess antibody in a population, you want to get a random sample of the population, as random of a sample as possible. And what I've understood about the New York survey was that it was done in grocery stores.
Yes.
And that there were actually, in some cases, reports of people showing up to the grocery store just to be tested because they wanted confirmation of a prior test. So that will definitely bias the results upwards in that situation. The Santa Clara study was a study that was recruited online through Facebook, and they did a little bit of sampling of particular groups, particular geographies, but their results are also coming into question with regards to the statistical analysis that they've done on the back end and the uncertainty estimates. And I think one thing I just want to mention quickly is because we don't know how accurate or reliable the antibody tests are, it could be that a lot of the positives that are coming up as positive in these situations could actually be false positives. And so this has to do not only with the reliability of the test, but also with the general prevalence of antibodies in the population. So there's a lot of discussion in the epidemiologic community about which way the bias goes. In New York, I think while 20% seems reasonable to a lot of epidemiologists, given the burden of disease right now, it may be an overestimate given the way that the sample was recruited for that particular study.
Yeah, and going back to the question that you asked me at the very beginning, we also don't know whether these antibodies, the presence of the antibodies actually is correlated strongly with protection.
Right.
It's probably correlated somewhat, but how strong is that correlation? What degree of protection we can expect?
Right. Yeah, and I think that there's also questions about, does having symptoms mean that you're going to have a stronger antibody response than someone who's asymptomatic? And so there's a lot of questions that are yet unanswered. And I do actually want to say something about asymptomatic transmission, because I think that that's come up a lot. We have seen evidence that many people are carriers of the virus without actually showing symptoms. And this has really large consequences for transmission. So if people are not showing symptoms, they may not take proper precautions, they may not change their behavior. And this could result in significant transmission. And this is something that we talked about a lot in our HIV class, because that's what sort of happens with HIV for the asymptomatic period there, which is very long in that case.
Yes, yeah, and it's just a very different. Though thinking about HIV, one of the things I remember we talked a lot about is why people hesitated to get tested. Like when the test became available, a lot of people in the early 80s were reluctant to sign up for the test. At the time, it was a terminal illness without really a treatment, and also there's all this dread with the people not wanting to know. But like now we have like the opposite case where people are like signing up, lining up hours in advance in order to try to get testing. So there's been this interesting switch in sort of the mentality of wanting to know.
Yeah, and there's even people, I know you mentioned the challenge test for the vaccine. There's people signing up as volunteers for a challenge test, right? Yes. So you're seeing this massive amount of engagement with science, I think, in a way that we haven't seen before either, which, hopefully is a positive thing when it comes down to it. I think maybe perhaps one reason for that difference in enthusiasm about testing has to do a bit with stigma and the stigma that's associated with HIV. We did see quite a bit of stigma initially with coronavirus that was really manifested in anti-Asian rhetoric, anti-Asian behavior, initially in the early days of this outbreak here in the United States. And I know that there is still quite a bit of stigma around coronavirus, but I think, when we think about HIV, that's sort of a disease that's held on to stigma despite, however 20 plus years of presence in the United States.
Yeah, indeed. And I think continuing to think about the way that coronavirus and HIV have these, some parallels. Like what I worry about a lot is when this virus really takes hold in other parts of the world who don't have healthcare systems that are necessarily quite as advanced, that are already really fragile. Yeah. When Haiti starts to get cases in real numbers, when lots of the Southern Hemisphere starts to get hit hard, I really worry. HIV, we saw dramatic declines in mortality in the 1990s in the United States and Europe, and those same levels of decline lagged, decade, 2 decades for other parts of the world. And my hope is that we can do much better this time around in terms of the testing, the treatment, vaccine becoming accessible on a global scale, not just the wealthier, more industrialized nations.
Yeah. And also thinking about how response is very different internationally than it is here in the United States. So there's been a lot of discussion about, well, what will happen when coronavirus hits the African continent? And, a lot of nations have said we can't do social distancing the way that other countries can do social distancing. And so thinking about cultural norms and how they come into play, to prevention, but then also how that influences eventual transmission in these countries will be quite interesting to see. And the hope is that perhaps we can, in some way delay, now all countries have it, but delay transmission enough that by the time this sort of hopefully gets there and becomes a large issue, the vaccine is available, or at least treatment might be available to lessen the severity of the disease. So I'm trying to keep up with the questions. They are coming in.
There's a lot of questions. There's a lot of them. There's some people who were mentioning the COVID-19 might have been in the US earlier than previously thought, right? In the Bay Area, they identified people who died from it before the Seattle people, who were initially thought to be kind of the first. And in relation to modeling, does that number, by pushing that earlier, change any of the data models that you've seen, Nadia?
Yeah, it's a good question. And I can't speak to the validity of those observations. I've heard some controversy around that first sample that was detected on February 6th. I think there's a lot of discussion that's happening around that. But certainly it would influence our parameterization of models. And I think it could influence timelines as well, because it could suggest that perhaps the virus was circulating much earlier. There are many more people who might have antibodies, might have already been infected with this. So it would certainly change the dynamics of these models. It would be curious though, especially given the current rates in California and the success I would say that California has had with social distancing and flattening the curve. It would be curious to see the validity of those initial cases and the timelines for those initial cases.
And then there's some questions about mutations. Someone mentioned that they saw something about 30 plus mutations of COVID-19 potentially. And
what? Sorry?
Someone mentioned seeing that there were potentially 30 plus mutations of COVID-19. And I think it might relate to those who have recovered and then are testing positive again. Is that an assumption I'm making, or is that, or we don't know anything really?
Well, the mutation rate has actually been really interesting. And there's, speaking of timelines, I was looking at a preprint that was posted online earlier this week that showed this really cool map using the changes in the viral sequence to then trace out of it, because if you track the sequence of the virus, you can then have it, you can build up a chain of transmission, or a likely chain of transmission, and then sort of model out then the spread just by following the sequences. The mutation rate is relatively small relative to HIV. HIV has a genome of about 10,000 bases. It's about a third of the size of coronavirus. And it makes, on average, about one mistake every time it copies itself. And it makes 1 billion copies of itself per day. And so it's accumulating mutations at a rate far, far, far greater. Coronavirus, on the other hand, seems to be quite a bit more stable. Now, will it, how it evolves and how does it adapt to us? Like, we'll see how that plays out, and whether these mutations play into reinfection is something we're going to have to watch. I mean, that's what happens with seasonal flu, and so the reason that we get sick with the flu each year is that we're being exposed to a virus that is mutated, considerably from year to year, and so when we put together a flu vaccine each year, the WHO and others will do their best to predict which strain and which sequence of the virus is likely to be prevalent 6 months from now, and then use that to try to protect the population. But because flu, influenza evolves so quickly, that means that you can get reinfected with flu again and again each year because of this accumulation of mutations. And part of what mutations do is it changes that antibody response. And so the antibodies is a very exact molecular fit. Like, the antibody fits in like really exactly. You can look at beautiful crystal structures showing just how perfectly matched the antibody is with its target. If that target just changes subtly, you lose that strong affinity, and the antibody will not be nearly as effective. I'm not sure I answered the person's question, but I talked for a while. And
I guess something you just said sort of triggered a thought in my head, which was about antibodies again, going back to this idea of the sero surveys. I just wanted to note that, you know, a sero survey that's done today is actually telling us something about infections 4 weeks ago, right? And so we have to be really careful about interpreting anything that's done today as an indication of what the state of the disease is today. It's really a historical look at infection. So I just wanted to put that in there because I think there were some questions about those data from Santa Clara in the question.
And then a couple of people have asked you, Nadia mentioned that vaccines weren't the only way
significant effort, public health effort. One thing is obviously a ramping up of our testing capacity. We should be testing many more people than we're currently testing right now. And I think alongside that ramp up, something that can be done is contact tracing, which is really manpower heavy. It requires a ton of manpower, but being able to assess who people have come into contact with while potentially transmitting and isolating those individuals. Again, not necessarily the most effective or efficient, but certainly could help in terms of reducing spread. And I say that because I think there's a lot of anxiety and a lot of discussion about sort of going back to normal, reopening things up. And I think we have to be really cautious about those discussions without acknowledgement of the significant efforts that would need to be undertaken on testing, both viral and antibody testing, and then contact tracing. And then also thinking about our healthcare systems and our ability of the healthcare system to keep up with the hospitalizations, to keep up with the demands of personal protective equipment and ventilators. These are all things that would need to be considered ahead of time. So a vaccine is not the only way out. It certainly is the easiest way out of this, but that doesn't discount sort of the process from here until a vaccine is ready to be used, the amount of effort that's going to need to take place to contain the spread of the outbreak of the virus.
Yeah. I think another tool would be is if we can get better treatment online faster, you know, safe, effective treatment, you know, it would enable us to ideally reduce mortality from the disease. And even if we can just make it such that someone who has the virus stays in the ICU half as long, or maybe never needs to go to an ICU, yeah, that really then changes the stresses that puts on the healthcare system. And today's class on biochemistry was about possible treatments. And so we fortunately know quite a bit about the coronaviruses, since the SARS outbreak in 2002, and then the MERS outbreak. There's been a lot of really intense study into their viral life cycles. We know a lot about how the virus replicates. We know the key viral proteins that are involved. And so then we can pursue a rational drug design strategy to try and inhibit those proteins. Again, it's going to take time. It takes time, but we have a head start on that, and it is possible that might come online before the vaccine, which then will lessen the burden of infection, which then can have these spillover effects. And so I'm hopeful, but I think one of the lessons that I try and teach in the HIV course is that is humility for science. Like, we've had the antiretroviral drugs that are possible to stop HIV since the mid-90s, and yet 1 million people a year die because of HIV, and which is, these are preventable deaths, and it has to do with healthcare system, with delivery, with access, with all sorts of social inequalities, and the way that people's health is affected by those inequalities. And so, I'm definitely pro-science. Science has a huge role to play here, and as a biochemist, I'm hopeful that my colleagues engage in this struggle. Is actually, I know Harvey Mudd alumni engaged in this. Recent alumni in graduate school working on compounds that might end up being possible treatments. I still do so with the knowledge that it won't be enough to just have the molecule. We need a way of getting it to the people who need it.
Absolutely, and I think I will just echo that to say that every bit of science that we do is taking place in our social, political sphere, right? And so I think we have to be cognizant of the fact that even if we do produce great science, that doesn't necessarily mean that it's going to be implemented. And I think I just want to mention here, because I think we would be remiss not to mention it, that there are huge disparities in who is being affected by coronavirus. And this has to do with sort of historical oppression, historical racism in the United States that's shifted the amount and the availability of quality healthcare to people of color. And I think it's really important to say that, because I think it's really easy to just stick behind the science and talk about perhaps the pre-existing conditions that these populations have, but without contextualizing that in the history and the social structure, I think we miss out on really important and necessary observations about the way in which society and science interact. So that's just a thought. I think I would have been upset if I hadn't said that during this.
Yes, definitely. I mean, when you think about what are the risk factors, people talk about obesity and diabetes, and then what people will often say is race is a risk factor. And there was a great talk at Harvey Mudd in February by Dr. Maybank, who works at the American Medical Association. And she spoke about how we shouldn't talk about race as a risk factor for these various diseases. We should talk about racism. Like that's the problem.
Right.
And these structures in society that we deal with that lead to these inequalities and these disparate health outcomes. And HIV shined a very bright light on this. I think we're going to see this again in COVID, and I hope that this is a bit of a wake-up call to maybe do some addressing of these disparities.
I hope so. So we're very behind on questions, Erin.
We have so many. It's been hard to manage.
Yeah.
I think we are almost done with our hour. It went very quickly. I kind of want you guys to both touch on what you hope we all learn from COVID-19, where you see hope for our country, our medical system, how we're responding, humans in general, in relation to this pandemic.
Sure, I can start, I guess. One thing I have some hope about, and it is actually quite encouraging, is the ways in which people have community in sort of non-traditional, non-expected ways. And I think it's been really inspiring to see that even if we can't be together physically, people are still finding ways to support each other. People are still finding ways to help each other. And I think it's also an indication of sort of the resilience that we have as hopefully a society, as a community. And I think the other thing I want to mention is something we've touched on again and again. And my hope is that this pandemic, despite sort of, unfortunately, the toll it's taken on our society, I do hope that it instills more confidence in science ultimately, that people have a greater appreciation for science, greater appreciation for many cases, and that hopefully will improve systems in the future. Hopefully that means we'll have a more robust public health system in the future. Hopefully that means we'll have continued scientific funding in the future. So I really do hope that this increases people's confidence in science overall, and their trust in science overall as well. Those are my two takeaways.
Yeah, I share both of them. I'm looking at my notes, and like those were exactly the things I was planning to say. But I think sort of echoing that, and just I would say that I hope that as so many things have been taken away from us, I think it helps that'll help us understand what's really important and what really matters. And, and that we, informing these connections that we've, these kind of weird Zoom call connections that now is the thing that we're doing, we have an appreciation for how important it is that we are connected and how interdependent we are on each other. And, I think how resilient we're going to have to be. I mean, one of the predictions of a lot of the climate change models is that we will suffer disruptions like this in the future. And this is going to be part of the world that, our current students are going to grow up living in, and learning how to be resilient, learning how to adapt very quickly when something unexpected happens. I mean, I remember in the AIDS class, like one of the things that we talked about was when the next big thing happens, are we going to be ready? And just because it's not a matter of if, it's really when. And so I think we're getting a chance to flex our resiliency muscles. And when we face future challenges, I hope we're a little bit more ready.
Yeah, and I will say on the point of readiness, something I've been thinking about a lot lately is how Harvey Mudd has prepared me for the role that I currently find myself in. And I think it's been really interesting to think about sort of all the experiences I had at Harvey Mudd with research, with public speaking, with engagement with various communities, whether that's on campus or off campus. And I still think very fondly of the ways in which Harvey Mudd valued both, obviously, the humanities and the sciences. And I think we're seeing the importance of both in the coronavirus response. It's not just enough to have the science down. We also need to be able to care for people. We need to be able to reach out to people. We need to be able to understand what people's needs are. And so for me, I've just seen the synergy in what Harvey Mudd is trying to do in my current work with coronavirus. And I will give a shout out to Professor Jacobson, who had a class on scientific communication. And I actually still use the book that he gave me in that class because I think it's so helpful when talking to people about science. So so many things I'm grateful for. And I think there's many ways that Harvey Mudd will continue to contribute to not only this, but to many other challenges that we'll face in the future.
Yeah. Beautiful.
Well, thank you so much, everybody. I apologize to everybody who asked questions that we were unable to answer. We are going to do our best to keep track of them, and if Nadia or Karl has the opportunity to answer them, even in short little questions, we will work on that and try to offer that as a resource for people. There is just so much uncertainty right now in a lot of the data and the reporting, and that I worry we don't have great answers to every single question that was asked. So we'll do our best. We thank you all for joining us. And I want to thank Karl and Nadia for taking time. I would like to thank some other people in relation to this topic. Everyone who has supported the Community Emergency Aid Fund that was set up in response to COVID-19. We've raised nearly $240,000 to support students, faculty, and staff during this time, which is amazing. And then lastly, I'm going to plug our next Mudd Talks event is next Tuesday with Porter Adams, class of 2018. He's going to be talking about cybersecurity these days. And so I welcome you all to head over to our online offerings. Link is in your confirmation email to see our full schedule of virtual events. So thanks again so much. Have a great day wherever you are. Thanks for participating.
Great. Thank you.
Thank you.
Thank you.
Bye.
Bye.